Hey everyone! Today we're going to be discussing DNA replication, transcription, and translation. Everything in this video is drawn, videoed, and edited by me, Joanne Jacob. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell's nucleus, but a small amount can also be found in the mitochondria. A molecule of DNA is made of two strands, which bond within themselves and form a double helix structure. These strands are made of four monomers called nucleotides, known as adenine, guanine, cytosine, and thymine, known by just their first letter. These nucleotides bond to one another in a pattern known as complementary base pairing. Specifically, A pairs with T and C pairs with G. In DNA replication, the enzyme helicase unzips the double-stranded DNA for replication, making a forked structure. An enzyme called primase generates short strands of RNA to serve as a primer that binds to the single-stranded DNA to initiate DNA synthesis. The enzyme DNA polymerase adds nucleotides to the template strands. Because the enzyme can only add in the 5' to 3' direction, it replicates the leading strand continuously. On the lagging strand, this replication is discontinuous, with short fragments called Okazaki fragments being formed and later linked together by the enzyme ligase. RNA, or ribonucleic acid, is a sister molecule of DNA. This copies a piece of DNA in the form of messenger RNA, which is one of its numerous functions. mRNA transfers instructions from nuclear DNA to the rest of the cell to create proteins. Unlike DNA, it is single-stranded and is formed of 4 nucleotides: adenine, cytosine, guanine, and uracil, with A bonding to U and C bonding to G. Synthesis of RNA is simpler than replication. DNA is unwound to form a transcription bubble. RNA polymerase II forms mRNA by adding complementary RNA nucleotides to form an RNA strand. Once a termination signal is reached, the enzymes detach, the DNA is rewound, and the mRNA is released. Proteins are the workhorses of the cell that play leading roles as enzymes, as structural components, and in cell signaling, just to name a few. They consist of an amine group, a carboxylic acid group, and a side chain, which is the most important to their nature. These proteins are created by the messages sent from mRNA. The genetic code uses mRNA nucleotides as letters. A word, or a codon, consists of 3 nucleotides, which is a triplet code. A codon specifies for an amino acid in a protein. There are 64 different codons, but only 20 amino acids, so this code is called degenerate. 61 of these codons are sense codons, which means they code for an amino acid. 3 are nonsense codons, which means they stop or terminate the synthesis of these proteins. tRNAs are short single-stranded RNAs of about 74 to 95 nucleotides. Each tRNA has an anticodon that is complementary to an mRNA codon. It is covalently coupled to a specific amino acid and is used in the process of translation to create polypeptides. A ribosome is a site of protein synthesis. It is composed of 2 different parts. The small subunit binds to mRNA, the large subunit helps bind polypeptides together, and the tRNA has 3 specific binding areas on this ribosome. The A is where it enters, the P is where the peptides join together, and E is where the tRNA exits. Now let's take a closer look at the process of translation. Once the ribosome has recognized a start codon on the mRNA, a small ribosomal subunit will bind to the 5' cap. An initiator tRNA carrying methionine will bind to the mRNA. The large subunit will follow, and the translation apparatus is formed. Now another tRNA carrying an amino acid will bind to the A site. A peptide bond will form between those two amino acids, and a polypeptide has begun to form. The polypeptide is transferred onto the new tRNA, and the old tRNA is ushered into the E site and exits. Once this process has repeated and a giant polypeptide is formed, a stop codon will be recognized by the ribosome. Release factors will bind to the A site, and all of the subunits will be released. Now you are left with the protein product of all of your hard work. Thanks for watching this video, and I hope you learned a little bit more about replication, transcription, and translation.